Welcome back, everyone. Today we're going deep into the world of polymer chemistry.
Sounds good.
And we're looking at a paper from the Journal of Liquid Chromatography and Related Technologies
Okay.
by Wong, Sweeney, Khurrum, and Shalliker.
All right.
It was published in 2002.
Okay, yeah.
And it was a paper you sent to me.
I did, yes.
Now, what really stood out to me was this idea of diastereoisomers.
Right.
I think it's a topic many people listening might know and follow, but maybe need a little refresher on.
Yeah, it can be a pretty complicated topic.
So why don't we start with just the very basics?
Okay, sounds good.
What is a polymer?
So, a polymer is a large molecule made up of repeating structural units
Okay.
called monomers.
Monomers, okay.
Yeah, and you can kind of think of it like a chain with many links.
Okay, so each link is a monomer, and then the whole chain is the polymer.
Exactly, and the type and arrangement of those monomers determine the polymer's properties.
Which is why there are so many different types of polymers.
Yeah, exactly, and they have such a wide range of uses, too, like plastics, rubbers, even biological molecules like DNA and proteins are polymers.
So when we talk about things like plastic, we're really talking about polymers.
Yeah, plastic is just a broad term for a whole class of synthetic polymers.
Interesting.
Yeah.
So let's get into this idea of chirality,
Okay.
because it plays a really important role in polymer science, right?
It does. It's a fascinating concept.
Yeah.
Chirality basically means that a molecule, despite having the same chemical formula and bonding as another molecule,
Okay.
can exist in two different forms that are mirror images of each other.
So like our hams.
Exactly. Your left and right hands are mirror images of each other.
Right.
But you can't superimpose them. They're not identical.
I see.
And these mirror image forms of a chiral molecule are called enantiomers.
Enantiomers, okay?
Yeah.
So that's when they're mirror images of each other.
Exactly.
But what about when they're not mirror images?
So when you have stereoisomers that are not mirror images of each other,
Those are called diastereoisomers.
diastereoisomers.
Yes.
And this difference between enantiomers and diastereoisomers is important.
It's crucial, especially in biological systems,
Why is that?
because the different spatial arrangements of these isomers
Yeah.
can really affect how they interact with other molecules. And these interactions are essential for things like enzyme activity and drug efficacy.
So even a small change in the spatial arrangement can have a big impact.
Absolutely. It can mean the difference between a drug being effective or even harmful.
Like in the case of thalidomide.
Exactly. That's a tragic example of how chirality matters.
Yeah, for those who may not remember.
Thalidomide was a drug prescribed in the 1950s for morning sickness.
Right.
But it turned out that one enantiomer of the drug had the desired therapeutic effect,
Okay.
while the other enantiomer caused severe birth defects.
It was a terrible tragedy.
It was, and it really highlighted the absolute necessity of understanding and separating enantiomers.
Especially in drug development.
Yes, definitely. So in this paper by Wong and his colleagues, they were looking at diastereoisomers of a specific polymer.
Right. Low molecular weight polystyrenes.
Okay, and they were using a technique called liquid chromatography to analyze them.
Yes. Liquid chromatography is a really powerful method for separating and analyzing mixtures of molecules.
And in this case, they used a specific type of stationary phase.
They did. They used something called Carbon-Clad Zirconia.
Now, I'm not familiar with that material.
It's pretty interesting. It has a unique surface chemistry that makes it really good at separating diastereoisomers.
So it's like a high-tech filter for molecules.
Yeah, you could think of it that way.
And what did they find?
Well, one of the most interesting things they found was that the elution order of the diastereoisomers on Carbon-Clad Zirconia depended more on the isomeric form than the molecular weight.
That's surprising.
It is. You usually expect molecules to separate based on size in chromatography.
With smaller molecules eluting first. But here, the shape and spatial arrangement were more important than the size.
Right, which suggests that there are some very specific interactions happening between the diastereoisomers and the Carbon-Clad Zirconia.
And this finding has some interesting implications, right?
It does. It means that Carbon-Clad Zirconia could be a really powerful tool for analyzing all sorts of complex mixtures.
Not just polystyrenes.
No, not at all.
So this research, even though it's focused on one specific type of polymer,
Yeah.
it really opens up possibilities for all sorts of applications.
Absolutely. In fields like drug development, materials science, environmental monitoring, all sorts of things.
That's really fascinating.
It is. It shows how fundamental research like this can lead to some really exciting advancements.
All right, well, we've covered a lot of ground here.
We have.
but there's still much more to discuss.
Definitely.
Let's take a quick break and then we'll come back and dive deeper into the implications of this research.
Sounds good to me.
So before the break, we were talking about how carbon-clad zirconia could be really useful for analyzing complex mixtures.
We were.
But the paper also mentioned some variability with this material.
Yeah, they did find some inconsistencies.
Can you talk a bit more about that?
Yeah, so Wong and his colleagues, they noticed that the performance of the carbon-clad zirconia varied a bit,
Okay.
depending on the batch they used.
Oh, so different batches of the material didn't always behave the same way.
Yeah, exactly. And that's something that's really important to consider when you're doing this kind of research.
Because it could affect the results.
Exactly. If you're trying to compare results from different experiments and you're using different batches of the material, you might not be getting a true comparison.
So it's kind of like using different measuring sticks.
Yeah, in a way.
And that could lead to some misleading conclusions.
It could.
So what causes this variability?
Well, it's probably due to slight differences in the manufacturing process.
Okay, so even small variations in how the material is made can affect its properties.
Yeah, and that's something that needs to be addressed if we want to use carbon-clad zirconia reliably.
So there's a need for more standardization.
Definitely. More research is needed to optimize the manufacturing process.
To make sure that the material is consistent from batch to batch.
Right, and that's especially important for things like drug development.
Because you need to be able to reproduce results.
Exactly. Consistency is key in any scientific research, but it's especially crucial when you're dealing with things that could affect people's health.
Right, so this variability issue, it's not a deal breaker for carbon-clad zirconia.
No, not at all.
But it's something that needs to be addressed.
Yeah, definitely more research is needed.
Now I want to shift gears a bit.
Okay.
And talk about something you mentioned earlier, microplastics.
Right.
You said that understanding polymer properties, including chirality, is important for addressing the microplastics problem.
It is. Microplastics are a huge environmental concern right now.
They're tiny particles of plastic that are polluting our oceans and potentially harming marine life.
Yeah, and they can even end up in the food chain.
So how does chirality come into play here?
Well, for one thing, understanding the chirality of different plastics could help us develop ways to break them down more effectively.
Also, like designing enzymes or bacteria that specifically target certain types of plastic.
Exactly. If we can figure out how to selectively degrade harmful plastics based on their chirality, then we might be able to remediate some of the damage that's already been done.
That's a really interesting idea.
It is, and it shows how something as seemingly abstract as chirality can have very real-world applications.
Definitely.
It's all about connecting the dots between fundamental science and the problems we're facing in the world.
And using that knowledge to find solutions.
Exactly.
Now, going back to the paper for a moment.
Okay.
Is there anything else about their findings that you found particularly noteworthy?
Well, I think the fact that they were able to achieve such good separations using a relatively simple experimental setup is pretty remarkable.
So it wasn't a super complicated or expensive process.
No, not really, which is good because it means that this technique could be more widely adopted.
By other researchers?
Yes, and by industries too, potentially.
So it has the potential to be really impactful.
It does.
Well, this has been a fascinating discussion.
It has.
We've covered a lot of ground, from the basics of polymers and chirality to the potential of carbon-clad zirconia and the challenges of microplastics.
We have, and it all comes back to the idea that understanding the fundamental properties of matter is crucial for addressing the problems we face in the world.
Absolutely.
Whether it's developing new materials, finding ways to clean up the environment, or creating more effective medicines.
It all starts with a deep understanding of the science.
Exactly.
It's amazing to think about how this research on separating these molecules could have such a big impact on so many different fields.
It really is, sometimes it's easy to get caught up in the specifics of a particular study.
Right.
But it's important to step back and think about the bigger picture.
Yeah, and this study really shows how a seemingly narrow focus can lead to some really broad implications.
Definitely.
So what would you say is the most significant takeaway from this research?
I think it's the power of fundamental research.
Yep.
Wong and his colleagues, they weren't setting out to solve some specific problem like curing a disease or cleaning up the environment.
Right, they were just trying to understand this basic phenomenon of how diastereoisomers separate.
Exactly, and yet their findings have the potential to impact all those areas and more.
So it's a reminder that investing in basic science is important.
Absolutely, because you never know where it might lead.
Sometimes the most groundbreaking discoveries come from unexpected places.
That's right.
And for anyone listening who's interested in learning more about this topic,
Yeah.
where would you recommend they go from here?
Well, I think the field of chiral separations is a really exciting one right now.
Okay.
There's a lot of cutting-edge research being done, so I would encourage anyone who's interested to dig a little deeper into that area.
So maybe look up some of the researchers who are doing work in that field.
Yeah, and see what kind of advancements are being made in stationary phase materials and separation techniques.
That's great advice.
Thanks.
Well, this has been a really enlightening deep dive.
It has been.
I always learn so much from these conversations.
Me too.
And I'm sure our listeners do as well.
I hope so.
So thank you for joining me today.
It was my pleasure. Thanks for having me.
And to all our listeners, thanks for tuning in. We'll see you next time for another deep dive into the world of science.